recording, and let's see. Oh yeah, in Micro 101, disaster. I stopped the recording, played the song, and then I'm just like 2 slides in, and people are frantically telling me in the chat, like, Dr. Adams, record. And then someone was on the microphone. They're like, Dr. Adams. I was like, oh, you have a question? They're like, it's more like, can you please record? And I was like, all right, fair point. So you can be that person too. It's way better than what happened last year. Someone waited like 10 minutes and then finally I recorded. So we are recording, and now let us dive in to today's class. We'll be featuring Harold Amos. He is a really sort of inspirational figure in the field of science. He was also a passionate educator and mentor who was born in Pensacola, New Jersey, and the second of 9 children. So a big family. His father worked at the post office. His mother's name was Eola Johnson. And he was enchanted with microbiology and immunology. I mean, aren't we all? At a young age. And this was in part inspired by a biography of Louis Pasteur that he had as a child. And he was fascinated that Pasteur experimented on goats a lot. So a lot of his experiments were done on goats. And for some reason, Harold really didn't like their family goat. So he was like, hey, like, what a great use for goats. Let's use them for science instead of them bothering me at home. So that is a sign that your inspiration from science or for science can come from the most unusual of places. And yes, in the chat, some people are saying no one likes goats. Totally fair. But Harold's interest in science did not stop there. He went to school at a time where schools were segregated, but he nonetheless graduated high school in 1936, attended Springfield College in Springfield, Massachusetts. And it was notable because he attended it on a full academic scholarship, which was actually very rare for African Americans at the time. And he ended up graduating summa cum laude in 1941 with a major in biology and a minor in chemistry. So many of us may have done a similar major and minor program. During this time, the war had broken out, and he actually spent some time in the war as he was drafted and went to England and France, including the invasion of Normandy. And then he returned from the war, where he graduated from Harvard with his PhD in 1952. And this was notable not just because it's a PhD, which is already a big accomplishment, but he was the first African American to earn a doctoral degree from the Division of Medical Sciences at Harvard University. So really a pioneer in his field for a number of reasons. His thesis project was actually in virology, even though his mentor studied bacteria. And that just gives you a sense of how diverse the fields were at that time. And he looked at the infectivity of the herpes virus using a plaquing technique on what is referred to as the chick chorioallantoic membrane. And this technique was only discovered or created in the 1930s, so a pretty recent technique at the time, and is actually still used today to generate influenza vaccines, among other experimental models for studying viruses. And you can see in this schematic here, which you'll probably recognize because it's in your book, that this is an example of the different injection sites you can use. The chorioallantoic membrane is up here with where you might inject the herpes simplex virus. And so this is, again, another direct connection to our class. He became a faculty member at Harvard and served as a faculty member for over 30 years. He studied a wide array of topics, including bacterial RNA, cell protein synthesis. He did a lot of work on how you optimize production of insulin in eukaryotic cells and a number of other topics, not all of which directly relate to this class, but certainly show that he was a prolific scientist. And then at the young age of 70, if I retire at 70, I mean, who knows, but that's up there. He retired, but then get this, he became the first national director for the Harold Amos Medical Faculty Development Program. Now, it wasn't originally called this at the time. They changed the name because he served for over 2 decades. And this program's mission is to increase the number of medical, dental, and nursing faculty from historically disadvantaged backgrounds. So really, one of the most inspiring things I found about Harold is that he pays it forward and he gives back to all those who have helped support and nurture his love for science and mentorship. And I think that's a really important lesson, that as we are mentored and educated, that we remember to give back and pay it forward to the communities and people that help us, and so that future generations may also benefit from that mentorship. So if you're interested, I encourage you to Google him. Yeah, there are a number of great articles written about him, and he was certainly an important scientist, educator, and mentor. A brief announcement before we dive into our content for today. There was a good point brought up at the Student Success Hours earlier today. The open book policy for exams does not mean that you'll be tested on everything. So I completely hear students when they say that this book is dense. There's a lot of information in it, and it is a challenge to cover it all. And so I want to clarify just a few things in terms of studying and approaches for this class. You should not take super detailed, extensive notes on every page in this textbook. That will be overwhelming for you, and it will honestly, I think, be something that is less efficient and not as helpful in terms of preparing for the test. Now, certainly if you're just fascinated by all the information, by all means, dig right in. But I would recommend reading through the chapter or the assigned pages, and then making sure that you are grasping the big picture concepts and the main ideas. You not need to start memorizing or understanding all the molecular details in each chapter, because simply put, there's just not enough time in each class for us to go over each concept. With that said, after we go through that chapter or the assigned reading, then I encourage you to make sure that you have a good grasp of the slides that we cover and the corresponding material in the text. So today you'll see that we spend some time talking about plaque assays and HI titers, for example. Those are key themes that I would expect you to have mastery on for the exam. And if you don't, to come see me so we can work on that together. But there's no way we can talk about every single thing that's in the textbook. At the end of the lecture slides today, there are some that talk about intercellular membrane, proteins, and junctions, and we probably won't get to those, but that's okay, because remember, it's about the quality of your learning, not the quantity. And so please do not panic or freak out if you're like, I could barely finish reading this or taking notes on it. Do not take notes on everything. I recommend reading through the chapter, but then focusing afterwards on the key themes that we cover in class. So hopefully that allays the fears of of those who may have been a little nervous about the material, and you'll see that it does become more complex, but we do go slower through the chapters. The last thing I'll say is that open book does not mean you'll be tested on everything. So do not think that I am an evil professor who is like, ha, yes, I gave you open book. I gave you open internet. I gave you open notes, but there are so many different things I could pull from. You will never cover it all in 24 hours. No, like that's ridiculous. All I'm going to do is expect you to know what we focus on in class. That's it. So if we don't talk about it in class, then those concepts will not be on the test. That doesn't mean that I won't ask you to apply those concepts to maybe a new situation or something, but conceptually, what you're responsible for is what we discussed in class, not page 52 of a 30-page chapter that we never brought up. So hopefully that also makes sense, but if not, please let me know if you have any questions, and I'll pause to let you enter them in the chat or via the microphone, because I don't want people to be stressed out, period.
I have a quick question. So is it safe to assume that the reading quizzes that we've been doing are kind of like a guide for what we should be pulling out, like that kind of information to see it on the test later?
Right. Yeah. So take those reading quizzes, for example. They will definitely highlight some themes that I think are important, and those would then connect to topics on the test. I also would just caution that, I want to be considerate of your workload. So I'm not going to make a 10 question reading quiz or something like that. I'm just going to try to pick a couple topics that I think are important or notable, and then those will connect with themes in our lectures and exams as well. So you can use those as guides, but do not, yeah. So I'm not going to try to throw you off track or something. So definitely use those to your advantage. The other thing I'll say is that the more time we spend on something, you can think of that as the more time it'll likely be on the test, because I need to assess the students on something. So I want to assess them on things that we've spent time covering, not things that I mentioned for 5 seconds. Any other questions before we dive in?
Just to add on to that, so the level of detail that, for instance, with the ones that we've taken so far, would that be similar to what we would be tested on? Like the way the questions are formatted?
Yeah, so I would say yes and no. The level of detail is what I would expect, but I will not be asking multiple choice questions on the exams, because I don't think they're a good way to assess student learning. And also, I don't think it gives students a good opportunity to fully explain the level of their knowledge or the depth of their knowledge, right? For multiple choice, you're just circling an answer. In a brief, short answer, you get to explain the subtlety or nuance of your answer. And so that gives me an opportunity to award partial credit or even full credit, depending on what is put down. Whereas a multiple choice is a very black and white situation where I just have to follow, the answer key, right? And so unfortunately, I'm not able to do that on the quizzes. I just can't do all of the grading in a short answer format, but for the exams, at least I'm able to have that level of assessment. Okay. If there are other questions, student success hours are from 11 to 12. And also please let me know if you have questions via email. I'm happy to respond to that. My hand is getting, it's not fully recovered, but it's getting closer. So that's good. All right. So the key themes for chapter 2 are different methods for viral detection, techniques to measure viral growth, one-step growth analysis, and characterizing the cell surface, which we may or may not get to. first up, oh, right, and that specifically has to do with viral entry and exit. First up, we have the overall image or schematic for the infectious cycle. So we will have several weeks where we discuss the attachment and entry of viruses, as well as their ability to produce mRNA. Remember, that's the common goal that viruses must reach in order to generate their progeny, right? And so you have translation of proteins, you have the production of additional genomes, and then you have the assembly and release of the viral progeny themselves. And so the cycle begins anew. We won't be able to spend as much time on some of these intermediate steps just because of the constraints of time, but this first half of the class, we will definitely touch on some of these initial ones, some of the later ones, and then the second half of the class, we'll dive into the epic battle between viruses and our immune systems. That's also when we'll have a specific section on SARS-CoV-2. And at various points, I'll ask for your feedback if we want to make that shorter or longer, etc. So let's dig into these viral detection methods, and that is often done with cell culture. And so while cell culture is often at first glance thought of as music, media, and art that cells create, but in fact, we are referring to cells in tissue culture dishes. And for those of you who have worked in labs or had an opportunity to at least visit them, you've probably seen this in action to some degree. So this is a series of photos that give you some sense of the different dishes and techniques that are used in cell culture. Cell culture has a variety of flavors depending on the cell line you use. And this includes primary cell culture, which we can see here has an image of primary human foreskin fibroblast. There are also continuous cell lines, such as established lines. This is an image of mouse fibroblasts. And then finally, suspension culture, such as human epithelial cells or HeLa cells. Now, each of these have different pros and cons. So they all contribute to advancing our understanding of science and virology. And depending on what constraints you're navigating and the questions you're trying to answer, that will often dictate your choice of cell line. For example, primary cell culture has a limited lifespan, and so the cells will only propagate for 5 to 20 divisions, which in terms of experimentation, that is not a very long time. That means that you need to use the cells within a matter of days or at most weeks before they no longer are viable. An advantage, however, is that they offer very specific cell differentiation states. So primary cells are derived from either animal or human tissue, and they reflect closely the different cell populations, morphologies, and other characteristics that are most relevant to what they would look like within a living organism. They're also useful for vaccine production, and that also has to do with their close resemblance to living organisms. You can also consider the use of continuous cell lines, as opposed to those mere mortals, right? They have in terms of the primary cells, you have an indefinite lifespan. You want to live forever? Come join us, all right? That's a continuous cell line. They do have a uniform population, which often in science you think is a good thing, but here that's not great because you don't have that diversity, right? We hear how important diversity is in life, well, that diversity is important in science and your experiments too. So you may not get that true representation of all those different cell types that are present in a living organism. They are often treated with a mutagenic chemical or tumor virus to make them live forever, right? And this treatment will cause them to lose their tissue specific morphology. So they no longer closely resemble the tissues from whence they came, and or from which they came from, whence they came. So as a result, they can actually produce tumors, right? So when we think of cancer, we're like, oh my gosh, cancer is horrible. This is crazy. Let's destroy cancer. Cancer is simply cells that keep growing forever. So if it's uncontrolled growth and you have cells that have uncontrolled growth, they can actually produce tumors, which is a problem. One last upside is that they can help you synchronize your infection, right? Again, this consistency approach in science, that's usually a good thing because you can time everything, synchronize everything. And that means that you have synchronous infection and you can have more consistent results. Lastly, we have suspension culture, such as HeLa cells, and you can use a spinning magnet to continuously stir the cells. They allow you to grow large number of cells in a small volume, and this helps you generate large amounts of virus particles. And this is useful if you need a large quantity of something, then you can use that to either image, produce certain treatments, or you can apply X-ray crystallography, for example, and other microscopy techniques to try to assess your sample in a rigorous manner. So as you can see, they all have pros and cons, and what you use is often dependent on the question and your experimental constraints. An important note about HeLa cells: if you haven't read the book, The Immortal Life of Henrietta Lacks, I highly encourage you to. But this is a note about the respect for human privacy and confidentiality. So for those who are not aware, HeLa cells were derived from Henrietta Lacks without her informed consent. She was never told what they were used for, and informed consent for obtaining any human samples or participation is absolutely required. So while I hope you all go off to live successful and happy lives and careers, do know that should your career of choice involve the obtaining or use of human samples, you should always get you should always get their consent. And you should also always consider the ethical and moral implications of what you're doing before you pursue it. So if it is to serve the greater good and advance knowledge or treat disease, that is great. But make sure that you have those conversations with your colleagues, with bioethicists, with your institutional review board if you have one, and also with the patients or human donors that are participating, because this is part of them and they should be involved in these decisions. And this is my opinion, but I present it to you to consider. But I would argue that your DNA is your most valuable, tangible property. So something that you can actually see that's physically yours. And there are so many schemes and companies out there that are like, give us your DNA and we'll tell you what kind of, wine you like and what kind of, flowers best like fit your nose smells. It's like, are you kidding me? If you don't know like what you like, you don't need a company to tell you that. So like, don't give your DNA just randomly. Think about who these companies are and what they're going to do with their DNA down the line. So please please don't do it because you're like, I want to discover my long lost uncle's mother's cousin's brother. It's like, okay, like you might not, and now your DNA is in the hands of a corporation. So that is all I'll say there. Always happy to talk about that a little more in office or student success hours. All right, some general terms. Ooh, Gattaca, excellent movie. Definitely recommend. So in this textbook, there are different terms that are used. In vitro refers to experiments that are performed in the absence of cells. That's not the traditional use of that term, but in this textbook, that is how they use it. So that is how we will use it in this class. Ex vivo means you're experimenting with cells and likely viruses in culture. So like those tissue culture dishes I showed you earlier, that is what they're referring to. And then finally, in vivo means that these experiments are being performed in animals. Rarely in humans, those experiments are for the most part not allowed, but there are some exceptions. But those humans have given their informed consent. So, and yes, do not freak out if you did, 23andMe or whatever. It's not the end of the world, but certainly reflect on those choices moving forward. So the cytopathic effects produce damage to living cells, right? So this is an important concept. A lot of viruses, once you use them to infect cells, those cells will not be happy, right? That's sort of why we don't like viruses. And ways that we will be able to figure out that they're not happy are because these cells will change in ways that are visually obvious to us under the microscope. This can include a wide range of things, such as the size of the nucleus shrinking, the presence of vacuoles expanding or proliferating in the cytoplasm, the rounding up of cells, the creation of inclusion bodies, chromatin clumping together. There are a wide range of cytopathic effects. And I went ahead and pulled some primary literature from different papers. The links are on the bottom if you're curious in learning more. But you can look over time at cells and what happens to them at 0 minutes, 85 minutes, and 430 minutes. This is either uninfected or in cells infected with vaccinia virus. And you can see that maybe at the beginning, there isn't a huge difference in morphology, but with time, you can actually see quite a bit of shape change simply when you look at the shape of the membrane and the blebbing that has started to occur. So clearly this cell is not that happy. Likewise, you can use other microscopy techniques with vaccinia virus over a different time scale. Again, you can see that this cell is really going through some contortions here, not super happy about being infected. And this varies on the virus that is being used. So herpes simplex virus shown in the middle row, you can see that there are some distinct differences over time compared to the uninfected condition. Likewise, rhinoviruses, these are viruses that are responsible for the majority of common colds. I mean, look at 12 hours. This is all over the place. What is going on, right? So the type of virus and the cell type can lead to some very distinct cytopathic effects. Now, importantly, not all viruses cause cytopathic effects, which means that we will also need to consider those separately. One other type of cytopathic effect that is observable can be seen with Synctium formation by murine leukemia virus. And this is where you have viral glycoproteins bind to the receptors of neighboring cells, and they form this giant blob, right? So this is a more obvious cytopathic effect that you can see under the microscope. of all these cells just sort of joining this massive syncytium that is formed. Now, another fundamental technique that I alluded to earlier regards to how we propagate viruses. And so this was used by a number of researchers since the 1930s. And this schematic highlights the different ways in which you can inject and inoculate the eggs with your virus of choice. But to give you a little more in-depth view of this, embryonated eggs can actually propagate these viruses in the following manner. This link is shown here and shows you the engraving that is made, the inoculation site, the puncturing, etc. And then after the egg has been inoculated, you will wait for a specified amount of time, remove the shell, pull back the membrane, and then harvest the fluid which contains the virus. So while this can be a little graphic, I wanted to show you the individual steps on how this process is done. And I guess some researchers might say that it's a shell of a job, but someone's got to do it. Sorry, I know that was a bad pun, but I thought of it and then I was committed to it to see it through. A shell of a job? Are you kidding me? Hello, that's amazing. Okay, maybe it's not. All right, so let's move on to the plaque assay. So this is a fundamental assay when we think about quantifying viruses. So when we are quantifying viruses, we need a way to reliably measure the number in our inoculum. And this is important because that tells us how many viruses we were adding to our experiment. If we don't know that, then it's very hard for us to conclude make consistent conclusions from our findings. The plaque assay schematic shown here doesn't quite show all the steps, but I'll walk us through them one by one, and then we can discuss it in further detail. So what we have here is step one. You have a monolayer of cultured cells that are incubated with preparation of the virus to allow absorption. And so absorption is specifically referring to this moment when the virus encounters the cell and then attaches to the cell, okay? And then you have that infection step basically, and then you remove the viral inoculum from the monolayer cells. So you have a plate of cells, and then this leads to, you then add the virus, you then remove the virus. You then cover that plate of cells with a nutrient agar, and then that agar solidifies into a gel, which restricts the ability of the viruses to spread to infected cells. So you have a single virus, okay, and then that infects a cell, that cell lyses, or the virus spreads from that cell, et cetera, and keeps spreading to make a plaque, which is a zone of clearance that is reflected by these white circles here. And so you have these zones of clearance, and that will stop because you've put this agar on top, solidifies into a gel, and then the spread of the virus is pretty much immobilized. And so over time, the infectious particles produce a circular zone called a plaque, right? And if the infected cells are damaged, you can distinguish the surrounding monolayer. But again, this relies on visible damage. If the damage is not visible, then you're not going to really be able to observe that plaque. And this process can be repeated with 10-fold serial dilutions. That's what's illustrated here. of the virus stock. And so the virus titer can be calculated in plaque forming units by basically multiplying the number of plaques by the dilution factor. And ideally, this calculation uses a plate between 10 and 100 plaques. This is important, right? Because if you have too many plaques, then it's going to be inaccurate because it's hard to distinguish plaques from one another. If you have too few, then it's also inaccurate because this can be more sporadic, and a difference of 1 or 2 plaques can actually make a huge difference in your calculation. So based on your dilution and how many plaques you have, you can then back calculate to get your PFU per mil, or plaque forming units per mil. And through this, you're then able to quantify your actual virus titer. Are there any questions on this, or are we at a plaque of words? Nothing? A plaque of words? Lack of words? Nope. It's a tough crowd. It's okay. It's all right. I know not every single one of these is going to be a home run, or even it's not, might just be foul balls. That's okay. I'm here for I am here for the good and the bad and the viral. Okay. Any questions though? All right, great. Then we will move right on along. So next up we have the plaque assay dose response curve. So this is a way in which we can assess the kinetics of the viral infection. And the majority of animal viruses have a linear relationship between the number of plaques and the relative viral concentration. So you'll notice that the linear scale here will generate either a 1-hit or 2-hit kinetic relationship for your virus. For the majority of viruses, it'll be a 1-to-1 relationship. The more virus you have, the more plaques you have, and it is linear. However, there are some viruses, and viruses have all these special exceptions, and this is just one example of them, where the viruses have 2 RNA molecules that are encapsulated separately. So they have 2 special the genome is separated into different capsids, okay? And basically both of those genomes are required for infectivity and the propagation of the virus. And this structural difference results in a 2-hit kinetic curve where the number of plaques is actually directly proportional to the square of the concentration of the inoculated virus. Another way of saying that is, if this virus has 2-hit kinetics, you're not going to see a linear relationship. You're going to see this curve instead. And that is important because if you are simply looking at some data, right, the number of plaques that are present and then the concentration of virus, simply by graphing that information, you'll be able to figure out the type of virus you're dealing with. So if you're studying an unknown virus, then you could do a plaque assay, see the number of plaques that form over different concentrations, and now you have some information on the nature of the genome. And that can then tell you a lot more about how the virus can infect, propagate, disrupt the host cells, etc. simply by doing that assay. The limitation, again, is that the virus must form plaques, right? And so not all viruses form plaques because not all viruses form cytopathic effects or cause cytopathic effects. Examples of viruses that do not form plaques include some retroviruses, such as the sarcoma virus. And this basically has to be analyzed in a completely different manner. When cells are infected with this retrovirus, the infection results in cellular loss of contact inhibition. And if that sounds like cancer, it's because it's cancer, okay? Just this proliferation of cell growth. And what happens is where the virus infects means that there will be this growth of piles or foci, as they're referred to, instead of a single monolayer. So remember, when we're talking about cell culture, you have this nice confluent single monolayer. But then you have this retrovirus come in and mess everything up. It's like, no, we're going to build piles. I'm like, all right, cool, we're going to build piles. So this pile is referred to as a foci of cells that have lost all inhibition, at least contact inhibition, and they're growing on top of one another. And you can count these foci, and they're sort of like the opposites of plaques, right? There are areas where you have massive cellular growth on your monolayer instead of a nice single confluent layer. The term for counting these cells would be, or sorry, not the cells, the virus, are expressed in focus forming units, or FFUs. So let's take a pause here and think about some thought questions. This has to do with the nature of a virus infection and the ratio of the virus particle to plaque forming unit ratio. So let's put ourselves in the virus's shoes. I know viruses don't wear shoes, they wear socks, obviously. But if we were to put ourselves in these virus shoes, would you want to have a low, a medium, or a high particle to PFU ratio? And based on your answer, why would you want that? So take a minute, think it over. Do you want a low, medium, or high particle to PFU ratio? A higher ratio means lower effective. A lower ratio, high because one particle will mean too many plaques. Interesting. So let's talk about this a little further. So when we think about our ratio, I think maybe it'll be useful if we define this in a few more terms. So let's think about particle to PFU ratio in terms of what's possible. So a 1 to 100 particle to P, or sorry, a 100 to 1 particle to PFU ratio means that for every 100 particles, one of them will successfully form a plaque. If you have a 10 particle to PFU ratio, then for every 10, one will successfully form a plaque. So based on that, let's think about our answers. So some of you are saying low, so you don't kill the cells before you propagate. Some are saying medium. Some are saying high. They wouldn't want to damage surrounding cells, right? So that they can infect them and keep spreading and replicating. Interesting. Okay, so yeah, there's a lot of different ways to think about this, and there's not necessarily a right or wrong answer. This is just starting to get those wheels turning about what the pros and cons of having different particle to PFU ratios are. Carly's saying low because it takes less particles to form a plaque, so that's a stronger virus. Or some might say a more efficient virus. If you have to produce 100 particles, 100 virus particles to get a single plaque, that means if you only produce 50 particles from a cell, you're probably not going to get that plaque, right? So that's another way to think about it. How many particles are going to be required so that you can continue to propagate and exist in the world? So again, there's not a right or wrong answer, but there are different considerations for what means you're a efficient virus versus a inefficient virus, or what implications this has for how often you mutate, etc. Let's think about this second question a little bit. What is the lowest particle to PFU ratio you can have, and why? Miguel says 1 to 1. Are you sure, Miguel? 0 to 1. 1 to 0. Are we just typing numbers in the chat? I feel like we're talking in binary now. 0 1 0 1 1 0 0 1 1. All right, well, let's think about it. 1 particle to 0 plaques formed. Is that a ratio though? Is that a ratio, 1 to 0? Why not 0 to 0? So you cannot have half a particle. That doesn't make any sense. You either have a particle or you don't. You can't have half a plaque. So for definition, the particle is what was referred to also as a virion, or the virus progeny. So when a virus particle is released, that is a virus going off into the world and then infecting another cell. Okay, so that is the term that virologists use to define the virus going off and infecting. So is it particle to plaque? Yes, particle to plaque, exactly. And so to help give us some context, here are some of the examples of particle to PFU ratio. So we can see that some viruses, like the polio virus, their particle to PFU ratio is 30 to 1,000, right? And they can go all the way down to the Semliki. I don't know if I'm saying that right. Semliki forest virus, which has a particle to PFU ratio of 1 to 2, right? So you're never going to see a particle to PFU ratio of 0 to 1. I mean, that means the virus doesn't exist, or 0.5 to 1, right? It has to have an actual particle, and you have to have an actual plaque, or else the ratio is undefined. So the lowest particle to PFU ratio you can have is 1. The most efficient particle to PFU ratio you can have would also be 1, right? Because that means for every single particle you produce, you're getting a plaque out of that. That's amazing. That's an amazing efficiency. There's basically almost no particle that is going to waste. Now, the implications for this with regards to infection, right, become more complicated. Maybe you are destroying all the cells around you and then you run out of cells too fast, right? So it could be advantageous to have in terms of not alarming the immune system, to have a higher particle to PFU ratio. But when it comes to efficiency and when it comes to what is low as possible, the ratio is 1. Any questions on that concept?
So when we're discussing this this ratio, it's more in terms of just infectivity, right? Not necessarily how dangerous this virus is.
Right. So infectivity does not equate to danger. That is more about virulence, right? So, and there are also a whole host of other factors which can play into that. And that's an unintentional pun, but also a pun because host of other factors, right? Like what host are we talking about? What is a host immune system? I mean, you could say it's a host of problems, lots of fun puns there. But the danger of a virus has to deal with the virulence. So what virulence factors does it have? How lethal is it? How easily transmissible is it? That also, how can it be transmitted? Can it be picked up by touching surfaces? Can you breathe it in? Can you eat it? I mean, there's a lot, right? But when we're thinking simply from an infection standpoint and plaque formation, that's what this ratio is dealing with. Great question, Christopher, or I don't know if you go by Chris, but great question.
Either or, it doesn't matter.
All right, Chris/Christopher. Any other questions before we move on? All right, then let's move on. Oh, why might a virus have a high particle to PFP ratio? So we already touched on that. And I thought this image was super cool. We'll touch on this later in the course about how live cell imaging allows you to see viruses attaching to cells. Like, look how cool they are, just like floating along. What chance does a mighty cell have against these little invaders, right? I don't know. All right, let's talk about the hemagglutination assay. What did we say about the last question? Oh, basically that if you have a high ratio, that could mean that you want to throw out a lot of decoys, maybe to distract the immune system. And that could allow your successful ones to sneak on by. So if you're producing a bunch of particles and your immune system is wiping out a bunch of them, it's okay because a lot of them could be decoys, or it could increase your chance of mutation, things like that. All right, so now to the hemagglutination assay. So this is a very common way for detecting the influenza virus, which contains proteins called hemagglutinin that bind to a receptor on erythrocytes called N-acetyluramic acid containing glycoproteins, rolls right off the tongue, and can be detected using this assay. So this binding can result in multiple cells forming this lattice formation or clumping. And we can visualize this as shown here. So in sample A, all right, this column, there is no virus present. So the erythrocytes just fall to the bottom of the well, which is reflected as this little dot. And it's important to note that this well, while it's circular from a bird's eye view, if you look at it from the side, right, from a profile angle, it's actually pointed, right? It's like a triangle. So the red blood cells will slowly tumble down and form this dot in the center. Okay, so that's how they form in the center. It's not a flat surface. Now, conversely, sample B has virus, so the erythrocytes form this diffuse lattice, okay, and that coats the entire well, right? It's this diffuse lattice, they're all clumped together. And so you can see this lattice form because the virus is present. However, as we dilute out the virus, right? So we start with a high concentration and then we slowly dilute out the virus in these twofold dilutions. At a certain point, there's not enough virus that's present to sort of Velcro everything together. And eventually we'll see this red dot form. Okay, so my red dot is now covering the red dot in the well. Okay, so basically you now have the red blood cells tumbling down. So when we do this assay, and so this lattice is forming here, right? The highest dilution of the virus that does not allow lattice formation results in a dot. And the last dilution where hemagglutination occurs, so basically when we see this lattice formation, that's hemagglutination occurring. That's at 512, 1 to 512 dilution. Thus, this dilution is said to contain one hemagglutination unit or one HA unit. Or if you think it's funny, you say one Ha unit. Oh my gosh, I literally just thought of that. What a horrible joke. All right, so how many Ha units are in 1 to 256 dilution? All right, I don't know if I can keep saying that. That is ridiculous. I've been staring at HA for so long and I never thought to say that. So how many hemagglutination units are in this dilution? You can drop it in the chat or the mic. Haha. And I can write this in the chat. How many HA units are in the 1 to 256 dilution? Oh, this class loves pushing the boundaries. 0.5 for the PFU ratio. Now it's 0.5 for the 1 to 256 dilution. So let's think about this. Which one has more virus in it? This dilution, 1 to 256, or the dilution 1 to 512? Which one has more virus? 256, okay. So if I'm asking for how many HA units are in 256, and there is 1 in the 512, then would we expect more HA units or less HA units in the 256? I can't outsmart this class. You're just testing me if that 0.5 HA units silliness. Right. So there's more, which means there'd be double because there is double the amount of virus present. Thanks for keeping me on my toes. All right. So that means the virus is increasing in HA units or Ha units as we move up. And if anyone said they're haha, then technically, right, that's 2 HA units. So you're sort of right, but careful about putting that just on the test. So I now have another question because I've gotten a little confused, but if high dilutions of the virus prevent clumping, then why does this also occur at the 1 to 4 dilution? So why do we see a red dot at the 1 to 4 dilution, but we don't see it in any of these other ones until we have too little virus? Why do we see a red dot at 1 to 4? It's right there, and you see it? It's right next to my red dot. Too concentrated with virus right. So the red blood cells just freak out. That's actually correct. And let me tell you why. Because hypothetically, if this were to come up on an exam, you might want to know. But we have this model, right, where we have the virus binding the lattice together. So we have more and more lattice. It's great. But when we zoom in on the virus, it's not all just hemagglutinin. There's also this protein called neuraminidase. And neuraminidase has a specific function, which is that it cleaves sialic acid host receptors, okay? Because hemagglutinin is responsible for helping bind the virus to cells, right? It's how the virus can attach and enter host cells. But the virus also would like to leave the cells at some point. So it goes into the cell, it replicates, produces these proteins, does all this stuff. And then it's like, all right, let's pack up, gotta go. But, you know, they've spent so much time with the cell, they're sort of holding hands or receptors. It needs a little help letting go. And neuraminidase will cleave the host cell receptors, releasing the influenza virus, and it'll go off into the world to infect all over again. The thing is, there's not nearly as much neuraminidase as there is hemagglutinin. Okay, so there's tons of hemagglutinin, not as much neuraminidase. And so only at very high concentrations is there sufficient neuraminidase to cleave and release these lattices from forming. So the lattice likely starts to form, but then is consistently disrupted and results in the red dot appearing. And so it is actually quite common, depending on the titer, for your virus to have this pattern where you have a red dot at the highest, but not at the lowest, right? But it also depends on how much of your virus titer there is and what dilutions you're looking at. So that is why it's important to think about how many HA units are present, as well as what dilutions you're looking at. So too much virus is the tip of the iceberg. This gives you the molecular details as to why too much virus actually leads to lattice disruption. All right. In the interest of time, we're going to move on to a variation on this assay called the hemagglutination inhibition assay. So here we do a slightly different addition of reagents or virus, etc. So in the prior assay, we were looking at different amounts or different dilutions of the virus. In this assay, we're going to look at one dilution of the virus. For example, one dilution of the 64 HA units. And there's a question in the chat about the previous discussion about hemagglutination. The correct answer to the question would be saying that the virus is so concentrated that neuraminidase is able to disrupt lattice formation. Simply saying the virus is too concentrated would not be specific enough on a hypothetical test question. The equal HA units are virus titer. It is equivalent in terms of concept here, are added to every well in a 96 well plate. And you also then have what you're diluting here is serum. So serum contains antibodies that can inhibit the influenza virus, right? So you add different dilutions of the serum in the wells, and then you add an equal amount of the erythrocytes to the wells. To give you a visualization of this, we have this image here, okay? So equal amounts of red blood cells and equal amounts of virus were added to all of these wells. What we're diluting is the serum from patient X who gave us their informed consent and agreed to participate in this study. All right. They aren't the 8-year-old boy that Edward Jenner was inoculating with smallpox. All right. So they donated some serum, and then we went into the lab. I know it's been a busy week. We probably don't remember, but we all went into lab. We diluted this serum. Okay. And at a certain point, we stopped seeing protection, right? Because early on, we're blocking the virus from forming the lattice. We're blocking the virus from forming the lattice. But then we've diluted the serum so much now that the lattice formation occurs. We then got another donor who donated some serum. And again, we see this series of dilutions, and we see protection again until we've diluted the serum so much that we don't see protection. And so this assay can be carried out in approximately 30 minutes, and the antibodies are what are blocking the influenza virus, okay, from forming these lattices. When we take the reciprocal of the highest dilution of the serum that prevents hemagglutination, which is referred to as the hemagglutination inhibition titer of the serum, or the high titer. I don't know why I never just decided to say these words as words. All right, the hemagglutination inhibition titer, okay? So this would be the titer of our antibody, okay? We can determine that simply by taking the reciprocal. So in this example above, the serum X has an HI titer of 20 because this is the last time we see protection. Once we get to this next dilution, we don't see protection. So we take the reciprocal. So a 1 to 20 dilution, the reciprocal of which is 20. Likewise, for the HI titer of serum Y, the reciprocal would be 320. So then the question is, which serum is more protective against influenza virus infection? Which serum is more protective? Why? I see a lot of Ys. Why not? Yes, Y is more protective. You have to dilute it more before it stops blocking the antibodies, right? So that would be the correct answer. To show you how broadly applicable this is, I mean, HI titers are used all over the research world. This is just a few articles I found in the PubMed database, in the course of a couple minutes, looking and using HI titers. So understanding how to do this is directly connecting you to research that is being done right now with scholars all over the world. And like scholars over the world, you are budding virologists, and we're going to put our newfound skills to use by helping this group that was looking at the cellular and humoral responses to influenza in Gabonese children living in rural and semi-urban areas, okay? So what this study did was they investigated whether these children from rural or semi-urban areas had different immune responses to influenza virus. So unfortunately, we do not have time to get into our research teams, but we're going to look at some of the data quickly and then pick this up next week. But what I want you to take a look at here is figure 1 from this paper, all right? And do not be scared of graphs. You're going to be so good at analyzing graphs and crushing the creation of graphs and figuring out flaws by the end of this class. You'll look back on these days and laugh like, ha, units. Okay. So what we're looking at here is the HI titer, right? We just learned how to determine HI titers. And what they are looking at are semi-urban and rural children HI titers, okay? And they're looking at days post-vaccination, okay? They look at day 0, 14, and 28 after they receive the influenza vaccine, and they're looking at the antibody titers against different influenza strains. So there are different influenza strains, and they look at these different time points. And so basically they're just looking at the HI titers from these individual children. You can then see the averages that are determined by these lines, okay? The solid lines correspond to the filled in triangles, and the dotted lines correspond to the open circles. What we can see when we just look at A and D, that at 28 days post-vaccination, there is a statistically significant increase in the HI titer of semi-urban individuals versus rural individuals. The asterisk indicates this significance. If you do not see an asterisk, then that indicates that it is not significant. So when we see this data, this would tell us that the semi-urban children have a significantly higher, statistically significant, higher HI titer than the rural individual or children. And so what we did, and I know, like I said, it's been a busy week, but what we did, just the other day, we all went into Duncan Hall. We got our samples out of the freezer, and we did these HI titers. So this is our unpublished data, all right, for 2 different people, all right, that were from this community, and we got their data for 0, 14, and 28 days from a semi-urban individual from the community and a rural individual from a community. And so what your task will be, and what we will continue with on Tuesday's class, is to see if our unpublished data supports or contradicts the findings from this published data, all right? Because we want to contribute to the study, maybe do another paper together. And so in addition to the reading review for next week, I'm asking you to go ahead and determine the HI titers for each of these patient samples. You don't have to worry about the controls, okay? But you do want to look at these patient samples. And then we will, together as a class, decide if the unpublished data we have generated supports this figure or is in conflict with this figure. And if you do not have time to do it, I totally understand, but I guarantee you this will be good practice for any test questions that might come your way on this material. So I do encourage you to do it if you have an opportunity. If you do not, we will discuss it in class so you won't fully miss out, but it is a great practice question. So with that, we will close for today. I hope you are excited about all the new techniques we're learning, all the lab work we've already accomplished. I mean, budding virologists are crushing it. I hope you have a great weekend, and I will be posting these slides as well as the recording either this evening or tomorrow morning. All right, take care. Have a good one.
Thank you.
Thanks, Dr. Adams.
Thank you.